Thank you. Thanks, Alicia. And share a notes document. So we have some notes that you are welcome to ask questions in. I'll share my screen to do a little overview of it. So this notes document, there are different places with resources, and then links, and then we have a place where you can put your name, where your institution, where you're coming from, and then a little bit about you. We have some questions about what you study, what your specialty is. So you're welcome to put in any information and ask any questions there. In this document, there are also places where you can, we'll go over an overview or an outline of all the different activities, and then a place for questions and answers, or your preguntas. So you're welcome to also put in any questions there as they come up. We will have everybody on mute for the most part during this webinar. So you're welcome to use the chat as another place to ask questions. But otherwise, you'll keep it to the virtual notes document or to chat, and then we'll have some time at the end of the hour where we can go over more activity, more hands-on activity, and ask any questions. That I'll get started. So this again is the introduction to KBase as part of the ISME Latin American workshop that KBase is doing alongside COSMIC, the Colorado State Microbiome Network. And this is really just a warm-up to get you into KBase so that you'll be prepared for the workshop itself. We have a few folks here today. So you have myself. My name's Ellen Dow. I am based at Lawrence Berkeley National Laboratory, and I lead our educators program. We also have Alicia Wood-Charlson, who's the KBase user engagement lead and helps connect our science. We also have Meilin Scott. She has been our intern and has helped translate a lot of these resources and our narratives. Do you want to wave hi, Meilin?
Hola a todos.
Thank you. And then Servin, who's an organizer of the workshop, as well as faculty at the National Autonomous University of Mexico. And I see we also have a couple other of our instructors who will be there in Mérida in August. So Sophie, Josue, do you want to wave and say hello?
Hello.
Awesome. I'm so glad you're there. And thank you for repping KBase, Sophie. Okay, so what are we going to do today? We have a few different goals. The first part we'll do is an introduction to the platform, to the KBase platform. So it's very much an introduction. We'll talk about terminology and other resources. After we go through the slide deck on introducing KBase, we'll then have an orientation to the webinar, or to the narrative itself. So how you build your different workflows in KBase or Flupos. And then we'll have a practical activity. Again, a few different ways you can participate. We have the virtual notes document here. And then we also have the narrative itself that you can work in, and chat. So those are a few different places where you're welcome to ask questions. I know mostly I'll be speaking English, and we have plenty of translators on hand. So if something I say does not translate really well, we can call on the different folks who are supporting me. Okay, so getting started, what is KBase? So KBase itself is a platform for investigating systems biology and performing research. It integrates both data as well as enables people to integrate, to build their own workflows and collaborate, and helps people with publishing their research. KBase itself is funded by the Department of Energy Systems Biology Knowledge Base. or sorry, I'm so sorry. I'm the Department of Energy Biological and Environmental Research Program. And so we fit into the whole landscape of user facilities, such as the Joint Genome Institute and EMSL, as well as facilities that help and resources that help curate data and publish it, the National Microbiome Data Collaborative and ESS Dive. And then we fit in more of the data analysis and visualizations part of that. And so KBase itself enables our users to integrate their data to access tools that are all open resource, and then customize their analyses by combining the data itself and these tools. And then as you're building out your own personalized workflows, you can then share these with collaborators and publish your results. So a few different aspects of KBase itself. So you can import your own data. You can use a few different types of data themselves, such as sequencing reads, assemblies, isolate genomes, metagenomic data or metagenome assembled genomes, metabolic models, expression data, and more. And so there's a few different ways you can do that through drag and drop or by using a Globus endpoint. So that's for any data that's going to be larger than about 2 to 3 gigabase pairs. And within the functionality of the platform itself, it does help you detect the different data types. So you can go through and figure out what kind, if it's a FASTQ reads file, if it's an SRA read, in the diagram on the right-hand side or that screen grab, that'll show you what it will give you suggestions for the different data that you are uploading. And then of course, we also integrate public data. So any public data that is available through NCBI RefSeq or also through some of the Joint Genome Institute resources, those are also available within KBase. And so once you have your data on the platform, something you will be able to do is build your workflow with over 200 applications for data analysis. And these are within different topics. So there'll be resources for read processing, assembly, annotation, which will be where we're focusing on, especially in the metagenomic realm. And then there's additional tools for analysis, such as comparative genomics, sequence analysis, metabolic modeling, and so on. We do have an app catalog that we'll take a look at, which describe all the different resources for analyzing your data. And then for those of you who are savvy and have your own tools that you like to use to analyze your data, we do have a software development kit. So again, just to look at the landscape of KBase itself and how you can customize your own workflows, there are quite a few different paths that you can take and build your own personalized analyses. So it's really very much on the user-chosen exploratory pathway system. And what we'll focus on within the workshop itself is more of a metagenomics workflow. So the one toward the bottom of this diagram. And how you build your workflows are within the narrative. So this is where you're building out your workflow. And here is where you'll have your data, any markdown information about the workflow that you're building, as well as the tools themselves. So what this looks like is on the very top part in the dark green, we have the toolbar. So this is where you'll name your workflow, and you will save it and be able to share it. On the left-hand side, there is the data pane. So this is all the data that is within this particular workflow. And then below that is where all the applications and tools are that you can use to build your workflow. On the right-hand side is an example of how you could build out a workflow. So we have the markdown cell on the top. So this is where you can add in your commentary. Below that is a code cell. So you are able to add in your own code if you really want to. And then below that, we have an app cell. So this is the tool itself. And then on the bottom is a data object. So this is one way that you can visualize your data within the narrative. And so these workflows, you are able to manage the sharing of it. They are inherently, so the first time you share them, they are going to be private, but then you can manage the sharing. So you can share the workflow itself with particular people and give them view only, edit, save, share options. You can also make your workflow public. And in addition to sharing with individuals, you can also share with organizations. On this sharing and collaboration page, right below the managed sharing, there is managed static narratives, and we'll talk a bit more about using KBase to publish. So I mentioned sharing with multiple people through organizations. So organizations are ways that you can organize all of your narratives that you're sharing with particular colleagues and other KBase users. So these can be whether you are in a lab together, a larger project across institutions, or if you're teaching, you can share narratives in an organization. The organizations themselves, you have a few different components to this page. You have the information, so the name about the organization on top. And then you have a few different content tabs where you can toggle between about, so information about the organization, the narratives, so all the workflows that are housed within this organization, as well as associated applications. And then you can also control the different members. So as an administrator, you can allow different KBase users to join the organization and access these different workflows. I mentioned before that KBase can, you could share these different workflows with students. And so KBase has been used with educators and in classes, primarily using different data analysis workflows and templates. And so we do have several workflows that have been translated into Spanish. We have the genomics workflow that has been broken down into 4 different narratives. So it walks through the different steps of assembly, annotation, data analysis, and then comparison over the course of 4 different narratives. And we have similar modularized workflows for metagenomics and pangenomics. And we will soon have, or might already have, a complete transcriptomics workflow. And so this really just builds off of the KBase infrastructure. And then I mentioned those static narratives. So KBase itself, we are trying to enable folks to really share their data and make sure it's reusable and also findable. So one thing that you can do with the narrative itself is you can create a static or an static version, which is essentially an HTML snapshot that has all the data and all of the different applications that you've used within that workflow. You can register these particular narratives with a DOI that you can then cite within a publication. And we also have a collaboration with the American Society for Microbiology resource announcements for publishing. So we have different templates and resources available within the narrative, as well as a place for students who have published with using KBase. So a little look at the static narratives themselves. This is one example of on top the publication of that's from a microbiology resource announcement. So these are four different bacterial strains. that have been isolated, and they're sharing this data announcement. Right below the top is the KBase narrative, and we can preserve the workspace. So that 179975 is the workspace of the KBase workflow, and then we can have an associated DOI to that workflow. This particular narrative has 10 different users, and there are 4 unique narratives that have been attached together. So each of the different students who are putting the workflows together went through and built out the different isolates themselves, and then they have a single parent narrative that they're built from. I just wanted to recognize the KBase team. We are spread across 4 different national laboratories in the United States. We are based in Berkeley Lab, at Oak Ridge National Lab, Argonne National Laboratory, and Brookhaven National Laboratory. So now we've just taken a little peek at the narrative itself, or at the overview of what KBase is and some of the capabilities. Now what we're going to do is we're going to walk through the different narratives, how these look together. So you're again welcome to ask any questions or go into the Zoom chat as we're walking through that. So if you don't have a KBase account just yet, you can go to kbase.us, and this is where you can register for an account. Up here, you would click the sign up button, or if you already have an account, the login button. And we have a few great ways on how to go through and do this. So we have a documentation page, docs.kbase.us. And here it is all in English, but you can navigate to signing in and signing up. And we have a step-by-step sign-up guide. And this walks through going from that sign-up page, how to sign up for KBase. You can use your Google Gmail. You can also associate your workflow with an ORCID account, which I highly recommend because it's a great way to help track any of the data narratives that you might be publishing. And you can link it there. So again, we recommend to go through ORCID. You'll also need to agree to the KBase user agreement and data policies. And once you've done that, you'll have your KBase account. I want to pause real quick and see if we have any questions. Take a look in the. Okay, I don't see any questions there. And I don't see any questions in the notes document or in the chat. Okay, so I'll keep moving forward. Is the pace good for everybody? Give me a thumbs up, thumbs down. I know most people have their cameras off, but feel free. Ah, Miguel. Oh, no. That was a fast hand. Okay. Alrighty. So once you sign in to the narrative itself, it will take you to the narrative navigator. So this is kind of like your landing page within the KBase workspace. On the left-hand side, this is the tab for the navigator. This is where we are right now. Immediately below that are the organizations. Then we have the app catalog, followed by the search, jobs, account information. I recommend if you want to associate multiple Gmails, or you sign in with your ORCID and want to associate your Gmail or want to associate your ORCID account, you can go there and link more accounts, including your Globus if you're looking to share to move some really big data sets. And then we have feeds and then collections of particular data sets. Here within the navigator, you can go between your own narratives that you've created. If you haven't been in KBase before, you wouldn't see any narratives there. Then there are narratives that have been shared with you. followed by the tutorials, which I have selected here. So these are all example workflows that you're able to look at, and then you can actually open these different workflows, and they'll open in a view-only mode. So not editable yourself, but you can make a copy of it and create and use it to play with and make your own workflow from there. And you get just a little snapshot of what the workflow, what's in the workflow. So you have some information about the author, how many data objects there are, what type of data is present in the workflow, and then how many different apps there might be used. There's 32 different apps have been ran. And then we have just a look at the data itself, and then a preview, so you can see the markdown information about this workflow before we open it up. And then what we see when we open it is going to be very much the same thing, where we have all of that data. Right below that, we have the different applications. And then another really nice feature is the outline tab. So up here on the top, we have that toolbar. When we have the narrative toggled into the Analyze, on the Analyze tab, we can see the data, the different applications. You can also view your workflows themselves on the Narratives tab. And then the Outline tab, which gives a nice overview on what different data and information is within this particular workflow. So similar to the nice table of contents that Ben built out with the scientific background, importing the data, running quality control, an assessment, assembling reads, that information can also be found here, and you can use this to move through the workflow itself. Now, I want to orient you a little bit more with the narrative. And so you can open up a new narrative over here. You click on the new narrative button when you're in the narrative navigator. You could also click up here on the top to open a new narrative. But this will open a blank narrative, and then I can show you what that looks like in KBase. Okay, so when we have here, now we have our workflow itself, and this is a brand new workflow. So up here, we have the KBase toolbar, and on the left-hand side, we have the data pane and the apps. And this is where we can start to add data itself. If I click on the Add Data button here, I can add data to this workflow. When it opens up the data pane, you have my data. So this is all the data that I have within my workflows. I can also access data that has been shared with me, publicly available data, example data, and then I can import my own data. And yes, so can we upload example data from SRA, from NCBI SRA? Yes. So in the public data pane, you have some NCBI RefSeq genomes. So this wouldn't be necessarily raw reads, but we do have the genomes here. And then another way to import, if you have specific reads that you want to import, you have the import button, and you can go through and upload data using a URL. So from the import tab, going to update with URL, you can use a direct download link. And I believe I answered that question. Luis, can you confirm? I don't see Luis. Oh, I do. So Luis, we have a question on, or Meilin. Do you want to step in, Meilin? For the, for uploading example data from
Yes,
SRN?
yes, yes, yes. Mario, tienes varias formas de subir los archivos de SRA a la plataforma de KBase. Justo ahora Ellen lo que nos compartió es visualmente cómo se pueden hacer. Puedes hacer la carga con el uso de una URL. Por ejemplo, puedes entrar a tu base de datos, no sé, decimos que esté en NCBI. Puedes entrar a SRA y en el momento de descargar los archivos, en lugar de bajarlos, si no los quieres tener en tu laptop, por ejemplo, puedes copiar y pegar esta URL en el portal de KBase y en automático debe jalarte los datos. Entonces es una forma de optimizar el espacio en tu laptop y no tener que estar haciendo carga y descarga de archivos y manejo de archivos muy pesados. Puedes también usar la herramienta de Globus, pero tiene cierto límite perdón, es para archivos muy grandes. Y finalmente, si tienes los datos, por ejemplo, en un servidor tuyo o en tu laptop, puedes, pues, básicamente jalar los archivos directamente desde tu servidor o tu equipo a Cabase. No sé si con esto termine el ejemplo y la respuesta, Mario. Perfecto. I think it's done, Ellen.
Yeah, it looks like it. Thank you. And if you want to step in at all, you are more than welcome to as we walk through the different points of the narrative itself, since I've given the general overview and now we're going through and looking at all the different factors. So that is one way that you can import data. Just as an example for editing the narrative itself, I can go over here and I can delete these different cells. So I can go here and delete cells just to personalize this workflow itself. So that's going through and adding data. You can also put in Markdown here. And this is where you can use different Markdown language to write in question or add in information about your data. Again, I can go through and I can delete cells if I need to. So that is a Markdown cell right here. And then if I wanted to add a code cell, you can add code cells as well. So you can see as you go through and start to add data, whether it's public or private, and go through, I can add data. If I'm ready to close this particular pop-out, I can either hit the close button or I can click this little arrow. And then I can add data to this workflow. I want to look at a data object. And I see we have a question about categorizing and grouping in the notes document. Okay. So we have a question on if the narratives themselves can be categorized or grouped together. Yes. I can show one example of that. And we do have an example, I think that's been shown here. We can take a look at that. And here's an example of all of these different numbers are individual narratives. So they've all been grouped together. And then Louise answered about the NCBI SRA. And then for maximum file size, yes, for drag and drop, it's about, yeah, the 2 gigabase pairs and 2 gigabases. And then we also have documentation on uploading data itself. So when you're going through and you're trying to troubleshoot on adding data, we have a few different YouTube videos. So we have the different data types, importing data. So you can go through by the different data types themselves. You can also go through and downloading data. And then we have a little bit more information as well around using data and then specifically transferring data with Globus. Okay, so one way that you can go through and you can edit or run your analysis on your workflow is by taking, well, here I have a nice genome example, and I just did my first one, but I have a few different ways that I can go through and select from the apps themselves. So you can change the category, change the filter and how they're organized between the category, input type, output type. You can change the orders of the name. You can also go through and directly search. You can refresh it. And then you can also toggle between beta and released apps themselves. Another way, besides going through and looking in the app, examining and going through the app catalog itself over here, is to use the data object itself. So underneath each of the data objects, you have space where you have the showing apps with this particular data as an input, having this with the data as the output, exploring the data itself, going back in history, so reverting any changes, exploring the provenance, so how the data has been analyzed so far, and what you can download the data, change the name if you want to, and you can also delete it. But if I want to go through and look at the applications and see what apps this has been used in, I can go here to show apps with this as an input. And now I've already filtered for the different types of data that can be used. And so here, now I see that there's only tools for genomes that are available and accessible here. So this is the setup for the workflow and how you can build your own workflow. When you're ready to save it, you can click the save button. You can also share it again with manage sharing narratives, and then change the name of it if you're ready to save it. We do have an example workflow that we're going to go through today. And let me just go back and make sure that we don't have any more questions. Okay, we did have a data about sharing. Question about sharing data. Okay, but that's been answered. Okay. So now I want to lead you through an introduction example of using KBase itself. So here in the notes document, there is an example intro activity that you can click on and open. And Meilin, I wanted to pass it off to you if you'd like to walk through the activity for everybody. Oh, and then of course, I didn't update that link. I updated the bottom link. Let me go here. Or no, that's afterwards. Okay. Yes. Apologies. I did not update that that link. But here it is. So now I can change that. Okay. Mail break that link and update it. There we go. Now there's a Spanish version. Okay, Meilin.
Aquí es la introducción al uso de la narrativa y esta narrativa incluye una lista de recursos y enlaces útiles para ponerse en marcha dentro de KBase y junto con varias actividades para poner en práctica esta nueva información. Y la narrativa se refiere oh, no. I'm sorry, I think I'm having technical difficulty.
We can still hear you.
Oh, okay, sorry, I think my screen froze. Okay. So, la narrativa se refiere al espacio de flujo de trabajo en la plataforma KBase donde puede acceder e importar datos y realizar análisis para construir su flujo de trabajo. Y tenemos la introducción a KBase de grupos pequeños y tenemos una práctica de trabajo. En primer lugar, felicidades. Usted ha iniciado sesión en KBase y ha abierto una narrativa. Conozca la interfaz narrativa de KBase, cómo agregar datos y ejecutar una aplicación. Y también practique la edición de celdas Markdown. Es un tipo de celda donde se puede agregar y la actualización de la configuración de compartir para una narrativa. Y la actividad de seguimiento, miré este tutorial sobre cómo navegar por la narrativa para revisar la interfaz de la narrativa.
Okay, thank you, Meilin. So we'll use this as a point for an activity after the workshop, but this will be a way that you can go through and practice the different steps of going through with, we have like a lot of different resources here, and then you can go through and practice with adding data, choosing an application, and then Markdown within the workflow, just like we did over here. But before we'll get to that point, we've gone through the different points of the narrative with the navigator, the account. We've talked a little bit about organizations, but haven't fully gone into that. And we also haven't talked too much about jobs and the organization that we'll use with the workshop itself. So what we're going to talk about right now, what I'm going to go through is more on the organizations, and then show you where you can look at jobs. And then from there, we will stop for any more questions and further clarification. So within the narrative navigator, below or right within the narrative, if I go to the navigator, right below that, I have organizations. And this is where you can go through and search for the GROW organization. When you go into the organizations themselves, they are going to be on my orgs, and you'll want to make sure that you toggle to all organizations. Can you repeat that, Meilin, in Spanish for me? I might have. Oh, I've lost Meilin. Luis?
Yes. Can you repeat again the phrase you went to translate?
Oh, yes. When you go to the organizations, you want to make sure that you toggle that you go to the all organizations versus my organizations, because the my organizations is where it immediately goes.
Ok, básicamente, si ustedes quieren compartir estos datos o, por ejemplo, también sus narrativas, pueden hacerlo dando clic en el icono que tiene a las tres personas, que se llama orgs, y básicamente tienen que dar clic tienen que dar clic en el menú donde dice All Orgs, a todas las organizaciones. Tienen que dar clic en esa opción.
Thank you. And this is where you can then go in and search for the Genome Resolved Open Watersheds database. And this is also the link that Alicia put into the chat.
Sí.
So this, when you click on here, and you'll want to request to join the org, and this is where we will share the data for the workshop, and you'll be able to access that. So this is something that you will need to do. So if you do put in a request to ask to join, we can let you into the organization so you can then access the data during the in-person workshop.
I can help translate. Básicamente para la actividad que será presencial en el curso precongreso, una recomendación es descargar, o bueno, más que descargar, tener acceso a los datos. Y para la actividad se está usando un set que fue generado y que está disponible en esta base de datos que se llama Genome Resolved Open Watersheds Database. Entonces, básicamente lo que él nos solicita es que hagamos esta solicitud de tener acceso a esta base de datos, dando clic en el All Orgs, para que puedan tener acceso a estos datos antes de la actividad presencial. Entonces, básicamente el ejemplo que se va a hacer es usando estos datos, que por cierto va a estar disponible la persona que generó los datos para hacer preguntas de metadata y demás. Entonces, bueno, básicamente es la forma y el pasito previo que tendrían que hacer para tener acceso a esta información.
Thank you. So this is one place you can go. I can check if anybody has requested to join the org, and then you can just see a little bit behind the scenes on what that looks like. Yep, we have some requests. So again, within each of the organizations themselves, there is the about. So this is information about this particular organization. If you were to create your own organization for people that you are working with, you can edit this information. Here on this tab, narratives, these are all the workflows and data associated with the organization. And then we also have applications that can be associated. And then over here, you have members, our requests, and related organizations that we can, that have access, can look at all these different grouped workflows. Luis, could you go over quickly just about changing the about and the narratives and then managing the members of the organization?
Yes, Helen, I was writing on the questions and answers. Yeah. Can you repeat again, please? Just to be sure.
Yeah, Yeah, we're just going over the about, how this is the about is information of going over the organization. Narratives are all of the workflows and data associated with it, and then managing the members who are part of the organization itself, whether it's a class or a research group.
Ok. Bueno, básicamente cuando estén en esta ventana de orgs, de las organizaciones, pues la recomendación es explorar la plataforma. Pueden darle clic en donde está el icono de About y ahí pueden tener información acerca de la base de datos. Básicamente para todas las opciones, ventanas, bueno, programas que están como, yo lo veo como aplicaciones, pueden dar clic para tener la descripción, los links a los artículos, por ejemplo, o también las versiones de las bases de datos o de los programas. En el icono que sigue, que es el que se está viendo ahorita en pantalla, que dice narrativas, básicamente son los flujos de trabajo que utiliza KBase, que pueden estar fijos o que ustedes pueden crear, que es una pregunta que también estaba en el Questions and Answers, si hay opción de cambiar las narrativas. Bueno, a veces las puedes utilizar como un molde y después tú puedes hacer tus cambios, digamos, copiando la narrativa. Entonces, sí es ajustable, es como un template, me gusta verlo así, que se puede compartir con tus profes o con tus estudiantes y que después tú les puedes hacer modificaciones. Es como un template que puedes seguir y después modificar. Entonces, básicamente lo que estamos viendo en la pantalla son algunas narrativas que están dentro de lo que se tiene asociado a esta base de datos. Y bueno, finalmente el otro icono de apps es lo que les estaba comentando, que todos los programas o herramientas que están disponibles en KBase, lo bonito es que es como utilizar una aplicación en un celular. Entonces, básicamente tú solo seleccionas archivos, algunos parámetros y puedes poner a funcionar alguna tarea de bioinformática de una forma sencilla eh, y ver los resultados, ¿no? I think that's it.
Okay, awesome. Yeah, and going off of what I could understand from Louise with the different applications, you can, but once you go on more of an exploration through KBase itself, you can also look at all the different tools. So the read processing, genome assembly, these are all different applications that you can use to build your workflow. And
Okay.
then another item is going to be the jobs, but I'll pause for you.
So, básicamente lo que nos comparte ahora es que en KBase, cuando, por ejemplo, no sé, te dicen, vamos a hacer un filtrado de tus lecturas y tienes poca experiencia, o vamos a correr un programa de ensamble, o obtener un genoma a partir de un set de metagenomas, y no estás muy seguro de qué programa utilizar, puedes darle clic en Catalog, que es el icono que se está viendo ahora. Y básicamente los programas y las herramientas que están disponibles en KBase las pueden explorar de esta forma en Catalog, que es lo más sencillo. Arriba hay un espacio en blanco donde pueden hacer sus búsquedas de programas. Y bueno, básicamente lo ven así como se está viendo ahora en pantalla. Si le dan clic a alguno, Ellen, can you please click on anyone? Ah, Ahí van a poder tener acceso a la versión del programa y a una descripción detallada de lo que hace el programa, qué necesita de entrada, qué genera de salida. Y lo que les decía, que también pueden tener acceso a las referencias de estos programas. Y
Awesome.
en el icono de Jobs,
And here you can see I have not worked in KBase all week. So in the job browser here, this is where if you run into an error, or you can look at something's complete, what you can do is you can click on the little eye to look at details. And this is where you can find different errors for the job that you like ran into. And so if you run into an issue that you're having trouble solving, this is where you can go to look for what the problem was when you want to ask for some help. And I'll show how to go through that KBase help desk after this.
Ok, básicamente si ustedes quieren tener acceso a los registros de los programas que corrieron, tienen que dar clic en Jobs y ahí pueden hacer el filtrado de sus tareas por tiempos, de las categorías que ustedes hayan definido, o por ejemplo si algún programa tuvo algún error, pueden entrar, como ahora se ve en pantalla, a una opción como el log que utilizamos para ver específicamente qué tarea que tuvo de problema y así pueden hacer un análisis.
Awesome. So if you run into an issue and you do need to go here, again, going back to our KBase documentation site, we have troubleshooting. And then we do have a help board, and you will need to set up a Jira account. But we have a way on how to report issues and step-by-step instructions on how to create a ticket. And this will include, here's where you can include information about the issue. Like a longer description, you can tell us exactly what that error was, like we looked at before. Okay. My notes moved. Okay, so now you know where to join the organization, the GROW, which is where all the data is for the workshop itself. Again, we went through some details about, we've gone through jobs, as well as talked about the narratives themselves, the data pane, the apps pane, not so much an app cells and markdown cells. So in preparation for your introduction activity that you'll do, and then we'll also set you up with a little bit more on what you'll do next. But within your activity, it will ask you to run a genome assessment tool. So if I'm here and I have sorted through my applications, where I can go is to, let's see, I think I just wanted to do quality control. Let's see. I think of, I need to refresh my mind. Not this one. Check in. That's right. So in addition to sorting, you can also go through, I can remove the sort. So it's at all the different applications. And I can put in check M and then assess genome quality. And now I have an application cell within this workflow. Here's where I click. For this one's quite easy. I just have one option for the input, but it's automatically suggested the genome because it's going to suggest an assembly, a genome, or a bind config. I can look at some additional parameters. And again, there are options that I can change. And to run an app cell, I click run. If I do need some more information about this workflow, I can click on the information tab. And we just went over in the catalog, but this is similar information to what you see in the catalog. I go back to configure, and then here is where I can run the tool. Again, now it's going into the queue before it will go into run. This is another place where you can look at the job status. So this will give you, once it starts running, it'll give you information about the job itself and if there's an error. So here you can get through between the info tab and the job status tab is similar information to both the job browser and the app catalog within KBase. Do you mind translating that, Luis? And then I think we'll be ready for any questions and setting up for the activity.
Yes. Bueno, lo que estuvimos viendo es un ejemplo de CheckM, que básicamente es una herramienta que nos permite verificar, entre otras cosas, la calidad de los genomas que se tengan pues disponibles, ya sea de ustedes o que bajaron de las bases de datos. Y también les ayuda a poder recuperar genomas a partir de metagenomas, que básicamente son los famosos MAGs, los Metagenome Assembled Genomes. Utiliza varias opciones para hacer esto. No voy a entrar en detalles. Y básicamente lo que ustedes pueden hacer es, si tienen un set de datos metagenómicos, ya sea el que se utiliza ahora de ejemplo, u otros, por ejemplo, de su set de datos, pueden hacer la exploración en el catálogo si les interesa recuperar estos MAGs. Y básicamente lo que están viendo aquí en pantalla es con CheckM, que es uno de los programas más utilizados en la actualidad para hacer este tipo de actividades. La recomendación también es que hagan sus pruebas. Si es un set de datos grande, en ocasiones puede tardar un poquito más de tiempo. Entonces, la recomendación es que si tienen sus propios datos, quieren hacer pruebas, tal vez en la sesión de preguntas con Ellen y Elisha y el equipo de KBase, pueden llegar con preguntas más puntuales después de haber corrido su set de datos en KBase.
Thank you. Okay, so the next parts of the activity, we've introduced some of these, and so you'll need to make sure you set up your KBase account, join the Grow organization, and then we have the activity that Meilin went through with copying, editing, and running a tool in KBase. Again, this is where you'll go through this particular activity itself. So making your KBase account and joining the org, and then you'll have the opportunity to run through the introduction to KBase. I want to see if there are any more questions. If there aren't, we also have a narrative set up for adding your own data and preparing it for so many questions. We'll have a we do have a workflow, and maybe right before I go into the Q&A, I'll go through the steps just so we have plenty of time to answer any questions. So after the seminar, if you have your own data that you want to upload and start analyzing during the workshop, we do ask that you have already uploaded and imported the data to KBase, and then have gone through using the tools of both FastQC, so a quality control, and Trim-O-Matic to trim the raw reads, and then have assembled your reads. And there's an option as well to do a comparison of the contigs themselves. And so this resource is open here in the notes document. Luis, do you want to run through this activity just real quick as part of their homework
Sure.
before August 4th,
Sure,
3rd?
sure, Ellen. Bueno, básicamente la recomendación es que si están interesados en este workshop, les recomendamos muchísimo que puedan subir sus datos en caso de que ya tengan datos metagenómicos. Ojo, no es con datos de amplicones, es con datos de metagenomas. O también si tienen genomas, la tarea puede funcionar. Entonces, la recomendación es que cuando tienen este tipo de archivos grandes, puede tomar un poco más de tiempo. Entonces, lo ideal es que hagan esta actividad como tarea para que también se familiaricen con la plataforma. Y bueno, básicamente esta es una narrativa traducida al español que les permite cargar sus lecturas, así tal cual se obtuvieron de un equipo de secuenciación, hasta recuperar genomas ensamblados a partir de metagenomas. Entonces, la primera parte es una introducción, viene allí el índice. Por cierto, si alguna de esas tareas ya las conocen, pueden darle clic a cualquiera de los números, 2, 3, 4, y los manda a la sección de la narrativa que les interese. Entonces, básicamente se divide en 4 partes, es la carga de sus lecturas, controles de calidad, la generación de un ensamble y básicamente hacer la comparación de sus ensambles. Entonces, para algunas personas puede que esto ya sea conocido, es una actividad, digamos, que puede tomar algún tiempo, entonces por eso les recomendamos hacer esto de tarea. Pero si es la primera vez que están haciendo este tipo de actividades de informática, los lleva paso por paso. Entonces, el primero es la carga y el importe de sus lecturas de secuenciación. Ya durante este webinar hemos visto algunas opciones para subir sus archivos con URLs, con archivos en su servidor, en sus laptops, etc. Y can you go down, Ellen, please? El segundo paso son controles de calidad. Básicamente, recuerden que siempre es necesario, aunque su proveedor de lecturas les diga que las calidades de sus lecturas están bien. Bueno, nosotros siempre les recomendamos hacer una verificación de las calidades y básicamente KBase tiene varias herramientas para revisar calidades, hacer cortes por calidad, quitar adaptadores. En esta tarea se les recomienda utilizar FastQC, que es uno de los programas más utilizados para revisar las calidades de sus lecturas y hacer edición. Y en el tercer paso, que es la generación de ensambles, pues es básicamente la tarea, ahora sí, de tejer sus lecturas para empezar a obtener sus ensambles, ¿no? Que es una tarea que todo el mundo quiere hacer como primer pues, paso, ya pasar de esas lecturas a tener contings, que son fragmentos de secuencia más larga que se obtiene de haber comparado, contrastado y unido a sus lecturas, ¿no? Entonces, en ese tercer paso se les recomiendan usar al menos 3 programas de ensamble y en el cuarto paso, que es el que estamos viendo ahora en pantalla, es hacer una comparación de dichos ensambles para, pues entre otras cosas, pues determinar cuál es el ensamble metagenómico que les va a servir más para sus tareas posteriores, ¿no? Como pueden ser la anotación, recuperar genomas a partir de estos metagenomas, etcétera, ¿no? O buscar genes que sean potenciales, no sé, blancos para alguna cuestión que a ustedes les interese muy puntual, quiero buscar genes de fijación de nitrógeno, etcétera. Entonces necesitan tener ensambles y pues el detalle es que si tienen varios ensambles a partir del mismo set de datos, porque cambiaron parámetros y o porque utilizaron diferentes programas de ensamble, luego viene la tarea de informática de decidir con cuál ensamble se quedan. Entonces en este cuarto paso les pedimos que hagan una comparación de sus ensambles utilizando herramientas que están disponibles en KBase. Y bueno, una de ellas es una aplicación parecida a Quast, que entre otras cosas les va a dar los parámetros que se ven ahí en pantalla, el número de contings, cuáles son sus contings más largos, el N50, L50, etcétera. Y can you scroll down, Ellen, please? Si no están familiarizados con algunas de las definiciones de estos métricos, las pueden ver en esta narrativa. Y finalmente, como una actividad opcional, se les plantea la posibilidad de hacer un análisis taxonómico, digamos, de entrada básico, utilizando Kaiju, que básicamente es una herramienta que les permite hacer comparaciones con datos disponibles en distintas bases, por ejemplo, de NCBI, que básicamente hace comparación con secuencias de proteínas, y esto les permite hacer un contraste de sus datos versus los que están en las bases de datos para tener una idea de cuál es la composición de su comunidad microbiana. I think that's it, Ellen.
Awesome. Thank you, Luis. Yes, so this will be the activity that you do. So to wrap up, just on this section, we'll stay on. We have plenty of time for questions and everything that we'll go through next. But we've gone through just an overview on what the KBase platform is. Then we've talked about different aspects of the KBase narrative, so how you build your workflows. And then we've given you some options to practice in KBase, including the introduction activity that you can practice loading and running a workflow, and then where you can go through and also practice running an app. Okay, so after this, you'll want to work on uploading and importing your data and preparing your data. One thing I did not show you is how to make a copy of an application. So before I do my final wrap up, I just want to go through and demonstrate how you can make a copy of a workflow. So here, this is Ben's workflow. It's not mine. I don't own it. So I would need to go over here and make a copy because it's in view only mode. So this is a way that then I can go in and have an editable version, like I have an editable version here. There's another way to toggle between view only and edit. So here in this view only version, I would go up here to make a copy. Can you translate that real quick, Luis? Just with the making a copy. Just because I see one thing about permissions as well, about accessing a narrative.
Yeah, well, I think Paulina already sent the link.
Okay.
So
Okay.
yeah, but I think it's kind of important also if you allow me. Cuando, por ejemplo, tienen una narrativa que les comparten y no quieren trabajar directamente sobre la original, pueden hacer una copia de esta narrativa, guardarla en su escritorio, en su sección de trabajo, y después hacer los cambios. Pueden dar clic, por ejemplo, en el texto de la narrativa y editarlo, por ejemplo, para traducir, para incluir una nueva tarea, hacer modificaciones. Y bueno, la guardan y después la pueden compartir con la persona que ustedes quieran. So I think that's it, Ellen.
Awesome. Thank you so much. So to wrap up, we will, once we have gone through all the Q&A, we'll let you know that we've answered all those questions, but we'll have time after this to go through and make sure they're complete. And we will share the notes document with everybody. In addition to sharing the notes and all the other materials, including the slide deck, and we'll also have a recording, and we'll make sure that those are available on the website. So everything, slide deck, these notes, recording, links to all of the different workflows for you to practice in. We will send those out in an email to everybody. And we'll also post it on the website just to make sure that everyone can access it. With that, I think we can stop the recording and go through to answering any more questions, making sure people have their KBase account.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.